Hello, I am JFigueredo, and I'll be investigating the influence of Melampus coffeus, or the coffee bean snail, on the establishment and distribution of mangroves. So what are mangroves? Mangroves are trees that are found in tropical to subtropical areas around the equator, or between 30 degrees north and south latitudes. Mangroves play an important role in protecting the land from erosion and also providing shelter for many different species. In Florida, mangroves dominate the coastline, and the three most commonly found are the species Rhizophora, commonly known as the red, Laguncularia, the white, and Avicennia, the black. The snail being investigated in this experiment is the macrodetridivore Melampus coffeus, or commonly known as the coffee bean snail. These snails are pulmonate gastropods, meaning that they do have a lung, so they cannot become inundated for very long periods of time. This is commonly why they're found in the mangroves as they climb the prop roots to get out of the water. They feed upon fallen leaves as well as seedlings from the mangrove forest. During my research, I came across a paper that did mention that questioned the snail's possible influence in distribution and establishment of mangroves, and that paper was the Profit and Devlin paper of 2005. Within the mangroves, there is a consistent zonation that is seen. Red mangroves are commonly found on the shore fringing, whereas the black and white are found on higher ground and out of the tide during low tide, but inundated during high tide. I believe that the coffee bean snail has something to contribute to this zonation. My hypothesis is that while the coffee bean snail does feed upon all three species seedlings, they favor that of the red and the black over the white, not consuming the white at the same rate. This has been supported by other research as well. Remembering what we discussed about zonation, as well as the fact that the coffee bean snail is a pulmonate, my assumption is that there will be an increase in snails as you move onto higher ground towards the white and the black mangroves, and therefore the red and the black seedlings will be fed upon in higher quantities there. To answer this question, 16 total hardware cages will be placed on the forest floor in pairs. One of those cages will have snails as well as the seedlings. The other one will only have seedlings and no snails. The location of these cages will be determined by the mangrove composition in that area. The cages will sit for 1 week's time, where I will return after that week and the seedlings will be collected and brought back to the laboratory, where they will be observed for damage from the snail, and then they will be planted to watch for germination and establishment over the coming weeks. To complete this methodology, I will use the $150 to purchase these materials, which will build the cages and help me grow the seedlings back in the laboratory. The importance of this research comes down to mangroves' ability to sequester many important elements as well as nutrients from the water and the soil around them. According to the Smithsonian Marine Station, Melampus are the only direct consumers of leaf litter in Florida, and therefore, because of this, many of the nutrient cycling that occurs in the mangroves is dependent on the breakdown of the leaf litter by these snails. If it were not for Melampus, many of those nutrients would not be cycled back into the soil and the earth. Many of those are nitrogen and phosphorus, which are used to build many amino acids and proteins. And if they were not cycled back, it would take many years for them to break down while in the peat beneath the soil. I would like to thank you for listening.